What we covered last time and the parts about constraints and scale separation that are up ahead, it's clear to me that this is going to be the shorter bit of presentation. So this session is going to be lighter on that and hopefully contain more room for discussion and future plans. The purpose of all of this is, of course, to help us work on a revision and an update of the roadmap to whole brain emulation. And we're still in the early stages of that update. I'm very grateful for all of you who are here to help. I looked over the insights that we collected in previous discussions, which are great. There were some proposals to use evaluations where we look at whether original system and emulation break in the same way, how original and the emulation change to achieve new behaviors. There was a lot of interest expressed in hemisphere on-off studies, the evidence from the impact of prosthetics on subjective experience, reported patient experience in stroke and its treatment, testing for qualia changes, using studies with reversible protocols, question about which type of information theory one should use in treating cognition as a machine learning task and how to attach meaning to information or its processing. There was the importance of enabling multiple approaches for flexibility and robustness of the path forward. That was a great point that Logan made. Considering what sort of success criteria could be applied to MRI-driven lo-fi whole brain emulation efforts. That was one of Emmanuel's questions. Looking for shortcuts in testing emulations, attempting to test subjective qualia on animals with rewiring experiments. I'm still not quite sure how to think about that because they're animals, but we could discuss that later. And studying peculiarities like illusions as an indicator for criteria and possible metrics. So that was really nice to get all of this feedback. And hopefully today we'll have even more time for discussion. So we have two main parts that are still up ahead, and that's constraints and success criteria. And this next part is about the constraints that affect criteria and metrics. Start with what are constraints? Well, I have here a definition, the specific measurable restrictions, which limit the range of possible solutions to a problem or project. And they typically describe a maximum level for some undesired attribute. This isn't the only way you can describe constraints. I'm sure there are many others and better ones, but really the important thing is that it limits the range of possible solutions that make sense. Generally, the nature of a mechanism can introduce uncertainties, such as with regards to the probability of synaptic or neuronal firing, and it will imply a ratio of signal to noise and will relate to associated schemes, such as population activity that provides some level of redundancy. Besides the mechanisms themselves, broader environmental circumstances are likely to introduce interference, such as, for example, through electromagnetic fields, localized changes in temperature or oxygenation, just to name a few of those. And not only do uncertainties and signal-to-noise issues impose constraints on the precision or implementation detail that can be counted on to meaningfully participate in the operations of a particular neural circuit, the requirements of cognition do so as well. For instance, different circuits or brain regions must be able to reliably communicate and interoperate. And that means, for instance, that phase-locked rhythmic communication, as one example, may fare better across the interface than making full use of precision in analog computation, which could be subject to chaotic random walks. So just thinking of it from the perspective of the brain and what it's trying to achieve, how it's supposed to be able to do things by coordinating the activity of different regions is relevant here. Also, upon waking or after anesthesia, the brain has to be able to return to normal function. And neuronal networks can't fully ramp up population activity for higher throughput, lest the output be an epileptic seizure. And there are a whole host of these up and down regulating and attentional and filtering modulations to consider in multiple stages of plasticity, from sensory input to short-term memory to long-term potentiation and eventually to lasting synaptic changes. I'm putting all of this under the banner of types of neural coding and the manner in which that constrains reasonable metrics and criteria. I'm not sure that the term neural coding or neural codes is one that is very well defined or is used everywhere in the same way. So I hope that, that we'll get to some reasonable definition or that everybody understands what I mean. So let's see here. For instance, a population code requires that the spiking of individual neurons is insufficient by itself for signal propagation and computation. And it requires that there's a threshold in terms of the activity of one population that can be a successful cue for the activity in the next. A rate code, by contrast, implies that not only are the individual spikes at single neurons insufficient, there also has to be an extended train or burst of spikes for there to be a rate code even, or for that to have meaning. A code that relies on spatiotemporal patterns of activity will depend on some minimum cue size to achieve activation of an attractor, if attractors are involved, if you've got recurrence in there. And most often it will also need temporal spacing to discriminate between successive patterns, which, for instance, recurrent inhibition can provide. And it may also require that heteroassociative strengthening or strengthened connections enable a useful succession of patterns of activation. Neural circuits may need to operate in more than one mode, and may even need to do so contiguously by rhythmically separating time into modal phases, for example, for ongoing encoding and retrieval. We can get into examples of those if you want to. There are some that I used to model and study, so they're a lot of fun to talk about. As mentioned previously, phase-locked synchronization can place limitations on meaningful signal propagation where circuits or regions need to collaborate. By contrast, in specific instances, the neural code may depend on mechanistic features, such as dendritic computation, to provide something like high temporal resolution in binaural sound localization. Okay, so one way to go about this, there's a lot more that we could write in the updated roadmap about how to systematically explore constraints and their effects on metrics and criteria for whole brain emulation. To do something systematic like that, we could attempt to list experimentally testable ways to exclude details from model functionalization by determining which types of information are unlikely to be preserved under specific conditions. For example, how much can subthreshold activity matter when downstream communication can be shown to depend on population spiking or under natural noise conditions? Similarly, what is the precision of spike timing that can matter when activity is phase-locked to a brain rhythm, or when recurrent inhibition and excitation squeezes the spikes of a pattern together? So at a minimum, we might be able to collect a list of references in the existing literature on that topic. I think I'm going to leave this slide mostly for your own review at some point. What it is it's a summary of a bit that we did on constraints in a whole brain emulation roadmap themed workshop a couple of years ago. I'm not going to go back to that right now. Here's some examples of ways in which neural code constraints can help us put some limits on validation and error correction. Neural circuits often exhibit sparse connectivity, forming structured networks with selective synapses. Consequently, automated segmentation algorithms don't need to identify connections between every neuron and can instead focus on the sparse selective connections that matter for functional circuits, reducing the number of potential connections to validate. Now, I'm not saying that you can't go ahead and identify all of them, but this might help, especially with the validation process. When mapping circuits, knowing that certain types of neurons, for instance, excitatory pyramidal cells, predominantly connect to certain others, like, say, inhibitory interneurons, that narrows down the space of possible connectivity patterns, making that task more tractable. Neural circuits often follow a hierarchical or modular organization, and you can then approximate complex circuits by focusing on core functional modules. For instance, cortical circuits are often composed of repeating microcolumns, or canonical circuits such as the feed-forward, feedback, and lateral inhibition circuits in the visual cortex. That can simplify segmentation and circuit mapping, and you may not need to test or reconstruct every neuron in its entirety. Of course, you have to be very careful, as we discussed in the journal club about the Lapalainen et al. Paper. The reason I say that is because you could be tempted to kind of schematize all of that across the board for all microcolumns or mini columns, but that might not make sense in some areas where the differences between different mini columns or the differences within them are really important to the information that has been learned, that has been stored in there. Whereas, say, in the visual cortex, perhaps that's an area where you might be able to do that abstraction the same way they did in Lapalainen, because it's an area where, say, you have a capability, a cognitive capability that, even if it is more stereotyped in an emulation, you may be able to learn to work with that. Whereas if something is containing, say, particular memories, then that's a different matter. In any case, common modular motifs help with error correction. If an automated reconstruction shows an abnormal pattern that deviates from the typical circuit organization, you can flag that for review or reprocessing. There are excitatory, inhibitory, and modulatory synapses, and their locations, axonal, dendritic, somatic, carry functional significance. Automated segmentation can be fine-tuned to look for specific types of synapses. So, for example, excitatory glutamatergic synapses typically form on dendritic spines, while inhibitory GABAergic synapses form on dendritic shafts or soma. That reduces the search space for identifying synaptic sites and accelerates segmentation. By knowing which synapses are excitatory or inhibitory and where they are likely to form, it's easier to predict how signals will flow within a circuit, reducing the need for exhaustive testing of every possible synaptic connection. If, for instance, a purported inhibitory synapse is found in an unlikely location, like on a dendritic spine, then you could flag that as an error and correct it, or a possible error and correct that. Similarly, errors in segmentation that lead to impossible circuit configurations can be caught early based on known synaptic rules. Neural circuits follow predictable patterns of activation based on their function, which can guide analysis and validation. Certain neurons, for example, pacemaker cells in oscillatory circuits, have well-defined roles and firing patterns. You might use that to approximate the behavior of the circuit without needing to reconstruct every detail. For example, the neurons in motor circuits tend to follow stereotypical timing patterns, which limits the number of possible functional configurations to be tested. If functional data like calcium imaging or electrophysiological recordings is available, then you can compare that. For example, if a circuit is expected to exhibit rhythmic activity and the reconstructed model fails to reproduce that, well, then you've definitely found something that's likely an error. Let's see here. Different neuron types, like pyramidal cells and interneurons, have stereotyped connectivity patterns, constraining segmentation and mapping. Knowing the rules of connectivity for different cell types allows for targeted analysis. For example, in the hippocampus, certain interneurons are known to target specific layers or regions of pyramidal neurons. This limits the number of possible connections that need to be tested during circuit mapping. If a connection is found between cell types that are not typically connected, this can indicate a reconstruction error. Neural circuits operate within specific temporal dynamics. So synaptic transmission and integration occur on the order of milliseconds to hundreds of milliseconds, constraining the likely flow of information. For instance, timing constraints mean that only certain pathways within a circuit can feasibly transmit signals in the time available, limiting the number of potential pathways to be tested for their role in a circuit's function. I would caution again, though, that you have to be careful there, because sometimes, even if there is that kind of a delay, it could be that the information being transmitted now is relevant for something that's supposed to happen later. So there's an intentional delay. Timing data from electrophysiological recordings can be used to validate whether reconstructed circuits align with known time delays for signal propagation between neurons. Let's see, who was that? I saw something went ping, like a hand going up, but now I'm not seeing it.
Oh, that was, I answered the question.
Oh, okay. I guess that was in chat. Should probably keep that open too. Oh yeah, okay. That's just about recording. Fine. Yeah. Many circuits are conserved across species or within brain regions. And those sorts of circuits, for instance, the cerebellar cortex, the hippocampal circuitry, they can be validated against known structures from other species. Highly conserved motifs like cortical microcolumns or recurrent neural circuits might allow for generalizations in some circumstances. Neurons have distinct dendritic and axonal branching patterns, which can be highly stereotyped depending on the type of neuron. Characteristic shapes and trajectories help automated segmentation algorithms distinguish between different types of neurites. If an axon or dendrite appears to have an implausible geometry, say, for example, an axon crossing into another neuron's territory or an unusual bend in a dendrite, that could signal a segmentation error. Because dendrites tend to receive inputs and axons tend to deliver outputs, this functional polarization limits the number of possible pathways through the circuit, focusing circuit mapping on the directional flow of information. As we work on this part of the roadmap update, what we really need is we need examples to clarify how you identify constraints and how they affect reconstruction and functionalization. So what we could really use is examples from literature where experiments or simulations showed either one or both of those things clearly. I'm thinking, for example, of studies where a circuit was reconstructed both using compartmental models and integrate and fire models. Or conversely, studies that show how an abstract model of an oscillator circuit leaves out important functional detail that crucially affects overall system performance. And after this session, I'm going to propose that those of us who care deeply about all of this share important references. I'll be looking specifically for these, and we'll probably do some journal clubs on them. See how fast we're going? Now we're already into scale separation here. Okay. Oh, let's see if why is that not going? What's happening here? Come on. Okay. What is scale separation? Scale separation refers to the idea that in complex systems like the brain, different levels of structure and function operate at different scales, and both spatially and temporally, and can be treated independently of each other for modeling purposes. In the context of whole brain emulation, this can be crucial because it implies that to successfully emulate the brain, one might not need to simulate all levels of detail simultaneously. At some level, we can aim for substrate independence, as we're calling that. Notice that the appropriate level depends on the goals, for instance, our success criteria. Take the example of emulation between computer systems, say a Mac on a PC or something like that. As long as program output is the goal, so you want to get the same output when you're running a program, then the instruction level will be sufficient. But if timing or physical effects on the computer matter a lot, like say where it's heating up or something like that, then that's a whole other kind of emulation. The biological brain operates at multiple scales, and here's a list of candidate scales. And these were also mentioned in the 2008 roadmap document. So you've got molecular, synaptic, cellular, and microcircuit levels, or the meso and macro scales. This diagram here, this needs some work. It was meant to say something along the lines of different levels at appropriate scale separation in different regions and how they work together, but this is not ready, I think. You could approach the relationship between scale separation and success criteria from either end when you look at option 1 or option 2. Either the appropriate level for scale separation determines success criteria, or success criteria determine the appropriate level for scale separation. And I think we're mostly leaning into option 2 here. Substrate independence at the scale separation boundary implies that details beyond that level are functionally abstracted away. As was already mentioned in the 2008 roadmap, the brain has a hierarchical organization with different processes and structures operating at different scales, from molecular dynamics, synaptic interactions, neuronal firing patterns, to large-scale network activity. Scale separation suggests that these can be studied or modeled in isolation to a degree. And the roadmap discusses identifying appropriate boundaries between scales, such as molecular, synaptic, cellular, and network levels, and determining how processes at different scales influence each other. The 2008 roadmap suggests that finding functional boundaries between different parts of the brain is one way to determine where scale separation may be appropriate. For instance, neurons and synapses operate at the micro level, while whole networks might represent a higher level of organization. Functional decomposition allows the researchers to focus on dynamics within a given scale, as long as its interactions with neighboring scales are adequately modeled. Although identifying boundaries between scales can help manage complexity, it's also critical to understand cross-scale interactions. For example, large-scale neural networks depend on synaptic activities, while small-scale molecular events affect synaptic behavior. We can pragmatically approach determining which boundaries can be treated as independent for modeling purposes and which cannot, based on empirical evidence and the goals of emulation. And here are two examples where stated goals may permit abstraction. So, for instance, the stereotyped convolution of typical circuits that we saw in the Lapalainen paper in that journal club that we did in perceptual areas, but also perhaps in motor areas, or, for instance, high-level model abstraction in some areas where we think that retraining is acceptable. So, say, motor or sensor areas, or where reuse and remapping occurs frequently anyway, so where it seems that that region of the brain operates more as a sort of resource that is reused over and over again. And I'm thinking here, for instance, about many aspects of how the hippocampus seems to work. And here are three examples where the application level of success criteria leads to corresponding assumptions regarding scale separation. So relating the behavioral level to sensor motor, there you can make the assumption that, say, at that sensory motor level, if that's behavioral only, then the internal implementation is irrelevant. There's the molecular level to nanoscale mechanisms. So here, if we're looking at that level, then our assumptions are that deterministic and non-deterministic mechanisms at nanoscale affect the goals of whole brain emulation in some way. And then the spiking domain, for instance, and how that relates to threshold and membrane potentials. There, the assumption can be that all mechanistic influences, however they come in, whether this is through diffuse transmission or through whatever means, whether there's any kind of influence by temperature and electromagnetism, whatever else, everything eventually becomes a contributor to membrane potential, so that all of the information in spiking systems is mostly carried in the spikes and through the timing of the spikes. Clearly, our dive into neural code, information preservation, and consequent constraints is directly related to scale separation. One way to explore the connections between neural code constraints and scale separation may be by using something like the perturbation framework proposed in the Fakar et al. Paper from the Korting lab. This is a fairly recent paper, and I don't know how many of you have seen it. But anyway, the study highlights a significant distinction between what neural activity represents and what it causally contributes to behavior. The paper ties into a lot of the other commentary that the Korting lab has about methods used in neuroscience. For example, regions of the brain that display decodable task-related activity might not be causally relevant to behavior. What they do in the paper is they draw from game theory. The authors use the so-called Shapley value, a measure originally developed to assess fair contribution in cooperative scenarios, as a foundation for assessing neural contributions. But they point out that traditional Shapley values are static and don't capture the dynamic time-sensitive nature of neural contributions. The study explores the effects of downstream transformations, such as summing or nonlinear operations, on the relationship between input activity and causal contributions. And they show that these transformations can distort the contributions of individual inputs, leading to discrepancies between recorded activity and actual causal roles. Their perturbation framework is proposed as one way to better understand causal contributions in neural networks beyond relying solely on recorded activity patterns. In perturbation experiments, which I'm generally very much in favor of sensitivity analysis and all of that stuff, they systematically remove nodes and observe the impact on contributions. You can also see how this relates to lesion experiments, for example. They conclude that simply observing neural activity, whether in artificial networks or biological brains, may not be sufficient to quantify the causal significance of neurons for producing behavior. It underscores the importance of considering downstream transformations when assessing causal contributions, and they emphasize using multiple metrics and tasks to fully understand the causal roles of individual nodes in neural networks. Examining networks performing multiple tasks or incorporating more complex architectures may be a way to see how causal contributions evolve as information is processed across layers. And that reminds me a bit of some of the comments that we had on the Lapalainen paper, because of course they did not study what they were doing in multiple tasks, and they did not get into a more complex description of the architecture. Instead, they stereotyped everything. Overall, this is a challenge to the traditional correlational methods used in neuroscience and to such interpretations of large-scale neural recordings. That's really the main point that they keep harping on over and over again. And I think harping is a bad word for it. It's a very good comment that they're making. Okay, so again, as we continue to work on this update of the roadmap, we need plenty of examples to include and refer to. Those should be examples of studies in which assumptions were made about scale separation, even if that was done implicitly without an explicit reference to scale separation, and studies that clearly show how some choices have good outcomes and others that demonstrate pitfalls. So that way we can begin to build a roadmap that actually helps to guide future work in a more systematic way than, say, when every researcher relies on cherry picking from a pile of papers. And I know that researchers don't intend to cherry pick, but it's kind of natural if a single researcher or a group is going about the business of deciding what they're going to rely on, what they're not going to rely on. If you have this if you have a larger review, let's just say, that's why review papers are typically great, especially batches of review papers, or a roadmap, then you can maybe avoid that a little bit. Again, I think sharing references between us can be a very good start. At this point, we've reached the end of the presentation portion of this session, and we're going to get into the discussion part. My last comment is to remind why I decided to talk about constraints and scale separation as well in a session that was actually about success criteria. I hope that you remember a little bit about the last session. The constraints and scale separation are really about model building and the choices that you make at that stage. So after data has been collected. But obviously, that's where you're closing the loop and you inform the improvement of success criteria and their associated metrics for a satisfactory outcome. So really the reason to get into this is that you can see the consequences of your choices. And that's where everything closes up if you have the right metrics, the right way of determining whether you're getting close to something successful. Now, quickly, before we get into the discussion, I just want to take a moment to express my gratitude to everyone, and especially to those who've been involved almost weekly in the effort to drive this roadmap update forwards. People on here have been put on the slide because they've been putting in a lot of work in that time. Logan, Maria, and Anders are part of that group, and Charles has been helping out a lot in an advisory capacity. By the way, for any one of you who knows her from her many science videos, if you haven't seen it already, you might be interested to learn that Sabine Hossenfelder recently came out as pro mind uploading, even though she didn't use the word mind uploading, in that she stated her belief that while she was commenting on false data and studies on supercentenarians, she thinks that the true route towards human life extension is by digitizing our minds. Just, a pointer to how this is becoming more popularly accepted, even in the case of those who specifically try to create an image of being a serious scientist who critically and rigorously critiques and is a skeptic of many things in science. Okay, so that is that part. And now we're into discussion. And the first part here is called Consequences, Type, and Resolution in Data Acquisition. And I wonder what you think our understanding of success criteria can tell us so far about the sort of data acquisition that needs to be done for whole brain emulation. So I'm going to, let me just look ahead for a second so you can see the other one's going to be model resolution in that. And then we have a piece about lo-fi. So really, first, there's the question of data, and there's a question of model. And for instance, there are success criteria for which the only conceivable good validation metrics or the only conceivable feasible reconstruction and functionalization methods require temporal observation of molecular processes, or require full molecular annotation, or require in vivo recording of intra or extracellular potentials of every neuron, require complete measurement of all neurite morphology, require knowledge of every synaptic connection or every ion channel, or cannot achieve successful thresholds past some degree of measurement error. So just to stimulate the discussion, but again, the question is really, what could we say about the consequences of thinking about success criteria on data type and resolution, and how to go about deciding what data to collect or anything of that nature? And I'm wondering if you have some thoughts on that. But also if you have other comments, other thoughts about anything in the previous session or anything about constraints and scale separation, please feel free to say so. Yeah, go ahead, Yunsuk.
Yeah, I guess if we think about whole brain emulation, it might be too tough. So I think we should just initially just focus on whole neuron simulation or whole neuron emulation and see what is needed to emulate a single neuron sufficiently. Then that will answer many of these questions. So another really interesting aspect is that the neuron is always changing due to different influx of ions, some different genetic, some different genes might be expressed. So a key to emulating a neuron is to emulate this adaptive property. How does it change to the different inputs that it is given and so on? So I guess that's another very important thing. If you just think about whole brain emulation, usually it's just, okay, now here we have a exact copy and we want to emulate it. But what's really important is how would it change in the same way that it does in the real biological brain?
Okay, I have two questions there. One is about the change and the other one is about the single neuron focus.
Yeah.
So the first question I have about the single neuron focus is, you know how when you, let's say in AI, for example, where you have challenges that are attempting to, say, help with computer vision, and then metrics are developed where, say, you've got a challenge, you have to identify these many things, and can you identify what you're trying to look for? And because the data set is a certain kind, and because the challenge is given in a certain way, people who take part in that challenge begin to focus on those specific images or those specific thresholds and metrics, the way that you can win the challenge, more than, say, on the overall goal, because it's perhaps not broad enough and there aren't enough aspects to the challenge, in the sense that it's not forcing people to focus on the main goal. So they become focused on, it's like they're making automatic assumptions about what matters based on that challenge. So in this case, the challenge would be reproducing the single neuron. And if that's the challenge, then you're automatically making a number of assumptions about, or at least you might be making automatic assumptions about the criteria that you need to use, about the scale that's important, about a lot of things, because you're going to be, if you're publishing it and you're comparing with other papers that are modeling single neurons, there's going to be a lot of emphasis on, oh, did they include all of this, all these different things, and did they reproduce how the neuron is generating spike trains or its subthreshold activity, and the activity in, you know, the potential changes at different parts of the neuron, say in dendritic compartments and things like that. So the focus might be more on those details automatically, without really taking into account how this matters in a population or in a circuit.
Oh yeah, I see, I see. So that's why you brought up the scale separation thing.
Yeah, that was in part.
Yeah.
Well, scale separation is important for that, and it's also just generally important because if you cannot separate scales, then you have an extremely difficult emulation problem ahead of you overall. But yeah, it's mainly that. It's like you're trying to focus on the overall goal. So that's why in addressing this part of the roadmap, we were trying to figure out what are the actual success criteria. So what is it that we're trying to achieve with whole brain emulation? If someone is most interested in producing a very detailed model of neurons because they care a lot about understanding what's going on mechanistically in a neuron, or they want to understand how a certain lesion will cause a certain thing to happen in a specific neuron, or how a certain drug is going to affect that neuron, I can totally understand that. But if the focus is on how do you recognize motorcycles, then maybe if you're trying to model the entire visual system, perhaps focusing on the single neuron could be a distraction in some ways. You see what I'm
Yeah,
saying?
okay. So yeah, got it. Yeah, thank you for the clarification. So actually, there are two parts in my comment. One was about the neuron level, and then the second part was about the adaptation.
Yes. Yeah.
So
Yeah.
we want this whole brain emulation to actually adapt in the same manner as the original biological brain. I guess that's my
Yeah.
second part of my comment.
There's a similar, not the same kind of answer or response, but something related to that that's in my mind, which is when we say, is the emulated neuron changing in the same way that the original neuron is changing? One thing that comes to mind is a single neuron, the way it behaves and also the way it changes isn't something that I think we can predict perfectly. There's going to be, let's say you run the same input through the neuron. You start from the same initial state, you run the same input through it, you might get slightly different adaptations in each case because there are uncertainties and probabilities involved.
Oh yeah. So I'm not talking about the adaptation at the neuronal level, but let's say at the higher level, like memories or
Yeah.
some kind of let's say habit type of thing. Would it adapt in the same manner as the biological counterpart
Yeah. Yeah.
at that
So
scale?
I tried to, in one of the success criteria that we listed in this big collection that we made, one of the things that I contributed to it was that I was trying to focus on when you have learning or adaptation or change in the system, sort of the path that it takes and how it changes. You probably can't determine that precisely because it's not necessarily that. I'm not going to say deterministic because everything may be deterministic, but it may not be as predictable. But there has to be an envelope of something that is considered reasonable. So whatever the success criteria are that you apply to that when you're comparing the original and the emulation is going to have to look at the envelope of what is reasonable as a path for it to take in terms of adaptation. So you don't want nonsensical adaptations, like things that ultimately lead to cognitive deficits or something that seems completely out of character. But you need to allow for the possibility that perhaps, or you don't need to. But I think what I'm saying is more, we might be willing to accept that the original and the emulation might not adapt in exactly the same way, as long as that adaptation is within the realm of what one would consider characteristic. I know
Oh
this
yeah,
is
yeah,
so
yeah, yeah,
vague,
yeah.
and it's the vagueness that, of course, we're trying to get away from, right? We're trying to make the vagueness more concrete somehow. Yeah, any ideas that people have about that would be great. Right now, the only approach that I really have to making this vagueness more concrete is to actually do simulations, to really dive in and compare simulations with experimental outcomes, and then from that try to extract a more concrete way to describe things like the reasonable envelope or something, because it's very hard for me to just go in from the top and say this is how it should be. But that's just me, and you know, I have one way of looking at things, so I don't know. There might be some better approaches there.
Right. Thank you. I have some more comments, but I'll probably wait.
You can totally make them now.
Oh, okay.
I forgot to write up anything about my responses, but I guess that's what the recording's for. Yeah.
All right. So my second comment rather was about the scale. So I guess there's obviously we're just thinking about the perhaps the spatial scale, but where I guess I have seen less mention about the temporal scale.
Yeah.
And so I guess that's also very important. Yeah, that's my comment.
Yeah, I absolutely agree. Yeah. Okay, I'm just going to write that down to make sure everybody remembers this.
Oh, and also variation in the input, or the variation in the temporal scale. How does this emulation respond to that? For example, if you play the same music very fast or very slow, at what point is it not recognizable as a particular tune?
Okay. Yeah, so I try to also include temporal, but maybe it didn't come out quite as clearly. On the temporal scale thing, I think that's where the bit about constraints actually really applies very well, because you have processes going on in the brain where, for example, recurrent inhibition will cause the spiking of cells that belong to one pattern to end up compressed together into one phase of a rhythm very closely, more than they might be if they just wander naturally without that recursion. Both recurrent inhibition and recurrent excitation help to do that, as well as oscillations that modulate that, of course. So you have that, and you have phase locking. All of these things are things that contribute to a certain temporal scale, because phase locking almost gives you a clock cycle in a sense. Of course, phase locking is different depending on what mode you're in. Say, for example, if you're in a mode where there's theta, versus in, say, something like slow wave sleep, where you have this rapid reactivation of patterns. But still, there's going to be some scale there. So I think it's quite likely that you can reduce what scale you have to look at to some chunks that are bigger than, say, microseconds or whatever. Yeah, I don't know what else to say about that, but I do think that's very relevant. I actually think that's one of the main or principal contributors to something we might call a neural code, is the temporal coding.
Right. So that reminds me that in describing, for example, the connectome, I guess it's very important to determine what is the conduction velocity of each connection. So without that, I guess we cannot really have a high quality emulation.
Yeah, so the conduction velocity, I think that's really important, but I think there's a higher risk that people leave out much larger contributions that will constrain the temporal discretization. Because when you look at what people are doing, say, for example, when Philip Shue went ahead and took the fruit fly connectome and tried to functionalize it by dropping in integrate and fire neurons and picking the same neuron model for each neuron, he did this all on a network that was otherwise quiet. And by doing that, he wasn't including, for example, any rhythmic modulation that would affect the propagation of these activities. Now, I don't know if it's in any way similar in fruit fly, because as we know from the Lapalainen paper, I learned that basically most of that visual system is subthreshold and not spiking. So I don't even know how to think about that right now. But if I were looking at a spiking system, then I'd be very concerned if someone was trying to learn something about temporal resolution without including rhythms, or without including modulation, rhythmic modulation, because then you're probably going to end up with a completely different, say, resolution that you have to look at. You have many more sort of very fine-grained temporal differences that you might end up with. Yeah. Sorry, I'm rambling.
Right. Thanks. That's all I have.
Okay, thank you. And by the way, I think we're combining our thinking about resolution in data and resolution in models, because we already started talking about resolution in modeling here, and that's totally fine. We'll just do them together. So any thoughts that you have on how there are consequences to our data collection and to model building based on success criteria, or the other way around, where the choices that we make in data collection and model construction affect what we mean by our success criteria? But also just feel free to mention anything. Yes, Ken.
All right, it's a good thing you said that, because I'm going off on a tangent. I think I apologize. So I really like the focus on scale separation, because I think that that really just nails it at some level. The thing that people have a hard time thinking about, including myself. So let's say that I want to say something very controversial here. So, you know, if you're going to emulate somebody,
Mm-hmm.
and you're going to emulate, let's say, their visual system, and you can include within that memory systems and anything else you want. Okay, and you want to, if they look at a rose, at a red rose,
Yeah.
and the success criteria is that the internal qualia experience of seeing that red rose is the same as the original person would have had. How does that relate back to the neuronal firing at each level of the visual system? Because I am under the impression that you could change V1 completely, and if you allow later areas to have their same response, that wouldn't change anything. But there's a whole literature on I guess on that debate. So if there's some very, the scale separation seems to really demand a what is the top level that caches out in conscious experience. And I just want to know your thoughts on that.
Okay. When you said, how does the qualia success criteria, how does it relate back to mechanisms like neural firing? To me, that seemed like the path you have to take. So that question is the path you have to take in the first place if you're going to ask a question like, is the qualia the same? Because I don't know how to test qualia besides really thinking about the mechanisms behind it. If somebody tells me that they have a certain experience of the red of a rose or something like that, to me, that's not enough of a description of the qualia, because I don't understand what it means to say, oh, red is red to me. It's so warm. Unless in my head, I'm thinking, okay, so there's an association somewhere between this concept of red and warmth. And how does a concept of red even come about? And I'm thinking about, say, connections from sensory input to previous experiences to, it always has to be a connection of things, because one of the things we mentioned last time was that patterns of activity in the brain don't really have any meaning by themselves. Any set of neurons that fires is completely meaningless unless it's in the context of other cells, downstream, upstream, how they connect, relate, work together. That's the only place where meaning comes about, is in those associations, really, in the way that they influence one another, and then eventually behavior. And that behavior can include putting something in your working memory that gives you the realization that you just experienced the color red. So I don't know exactly how that should work or how you should think about it, because of course none of us know the right model for any of this. but it's the only way that I find myself thinking about things like qualia. Is, okay, of course, this is a process where we're associating a bunch of things. Yes, Yunsuk.
So I guess when it comes to qualia, if we think about things like red or the smell of roses and so on, it becomes too complex. So the best place to study, I guess, qualia is pain. So the good thing is that it's binary. Either you're in pain or not in pain. And there's a lot of studies in animals regarding pain, right? So when you poke an aplysia, it might have this kind of withdrawal reflex and so on. So just based on the behavior, it seems like they have this pain qualia, right? But I don't know, is that just automatic reflex, or actually are they feeling pain in a similar way as humans? But anyway, it's just binary. Either they feel the pain or not feel the pain, and it's just a reflex. So
Okay.
I guess we need to look into this kind of literature.
We probably need to think a little bit about our definitions or what we mean. I'm kind of curious for anyone here. So if you're comparing qualia and the sensation of pain, or the aplysia's ability to sense pain and respond to it, what's the difference for you between those two things? What makes qualia something different? What would you describe as qualia as opposed to being able to sense and therefore experience and respond?
I guess no one knows.
Well, I mean, there's theories of consciousness out there that that may be wrong, but they at least have an answer at some level, right? So higher order theories that are saying that unless you have a self model that is able to represent that there is an I that is undergoing that pain, then there is no self, there's no qualia, there's nothing like to be the aplysia, because it does not have that type of self-modeling ability. And how do we know that it doesn't have that self-modeling ability? Because it does not have the behavioral output that suggests it makes use of such self-modeling ability. The Again, this doesn't mean that it's correct. Although I would say that like a whole brain emulation roadmap will be judged, at least in part, on whether it gets to the real question of whole brain emulation. And so putting this off as a gee, we don't know, but we have a fundamental faith that if we just get it, the details of the neurons correct, it's a very circular problem for a whole brain emulation roadmap to say we are going to punt on the idea of what consciousness is, what qualia are, and we're still going to talk about scale separation.
I certainly agree. I think it's dangerous to just punt on that. I don't think we can just say, oh, forget about qualia for now. We're just going to study circuits and then, qualia will emerge or something like that. It's important for us to know what we're talking about to begin with. If we say we care about this, I'm not even sure whether I should care about it because I don't know what people mean when they say qualia. So you just said qualia is basically when you combine a self model with a sensation. A self model is a representation of what? Like, take Metzinger, he's got we're making representations of the universe, of everything around us, and we're making representations of ourselves as part of that, and we somehow determine which part is us because we have more control over some parts of it. But that's a model of like stuff, of us, of the universe. And then you've got pain that happens. And then what is the self model doing with that? The self model is only able to say, yep, I experienced pain or a strong level of pain or something like that. It doesn't have direct access to the details of that mechanism or anything. So then when we say we want to reproduce the qualia of having that experience, is it sufficient to have a system that builds a self model and it gets in that says here's an amount of pain? Does it have to be a specific association? Like some people are much more, I guess it would have to be like a characteristic might be that someone is particularly sensitive to pain, whereas other people aren't, or they find it a much more important thing in their lives than someone else does.
Well, I've met a lot of neuroscientists that just at least um, the implication of their statements are that qualia goes deep into which ion channels are opening. That you're not going to get the qualia of a red rose unless you get the exact firing details correct in V1. And I think we probably all agree that that's nonsense, because at some level we've already accepted that a compartmental model is okay, so you can abstract beyond these things. But if there's no principled way of saying we are saying that we are abstracting beyond these things because, then it opens up to the same type of critique that has been leveled against whole brain emulation from neuroscientists for a while.
Right, but it's those kinds of things like details of ion channels where I'm hoping that that's where you apply constraints of the neural code and reliability and signal to noise and all that sort of thing to point out why that can't be the determining factor. But that doesn't, to me, that doesn't really answer the question of what it is that people care about when they say they care about their qualia.
So I guess we can actually test what qualia certain people have. You remember this old internet meme from, I guess, at least 3-4 years ago, where they have a dress and it's kind of has a stripe pattern. So some people see it as white and gold, and so some people see it as blue and black. Right. So the same input, but the qualia is different. So I guess this kind of illusion, I guess I spoke about illusion last time, but this kind of illusion can be used to actually probe into this phenomenon of qualia.
Interesting. Yeah, Keith.
So, I mean, the idea we're kicking around is often called one of the behaviorist theories of qualia, where you basically judge it based on third-party external judgments, because what else are you going to use? So if a creature looks like it's acting in pain, so be it. If a creature claims to see yellow or blue, so be it. That is, sort of one of the things we kind of have to go with. I just think it's worth sort of acknowledging that this is all basically based on third-party like determination. I don't even want to use the word judgment, but it's sort of admitting that that's the best we can do. We can't do better than to kind of observe whether or not an organism behaves in a fashion that we recognize as conscious in a particular, or experience quality in a particular fashion. So are we hoping to do better than that? Are we hoping to impose a model like it has to satisfy IIT requirements or something? Or are we just going to say, we think this thing looks like it's in pain, so we believe it? Right? What's our take on it?
So, but what are you saying exactly? I'm not entirely sure that I'm getting what you're saying, because, so for example, if we're asking somebody about their experience, we're asking about something that they consider qualia information, something that is about their qualia, about their experience. You're saying that they are unable to report it, but they still claim to have it, or that there is something other than what they can report?
I'm just saying that. So the dancing qualia experiment begs the question of whether or not we can actually trust it, right? You ask someone whether they see a blue dress or a yellow dress, and they give you an answer. Then you emulate their brain, and you ask them again, and if they give you the other answer, you assume that the emulation somehow failed or something. But or if they give the same answer, that's like a confirmation that you've preserved the qualia, therefore it's a good upload or something along those lines. But the whole dancing qualia concept is that you don't know if your qualia have changed. Your perception of red could be red, and then 2 seconds later, your perception of red could be what it had been green before, but it's still red. But by definition, you don't know the difference. It's sort of one of Daniel Dennett's points. So it's funny that on the one hand, we're dependent on these reports of qualia, and on the other hand, reports of qualia are definitively meaningless. I don't know what the answer is. I'm just pointing out that if we're making a public document, are we making, is that, I'm sorry, are we doing, so, you know, if this is meant to be seen by the various consciousness researchers of the world, they're going to see these things. And if we don't want to look like amateurs, we have to speak that language.
Yeah, I think the thing I'm having a hard time wrapping my mind around is how to speak about it, because I've tried to learn more philosophy by reading up on what some of the philosophers write about our experiences. But I'm still having a very hard time putting my finger on what they really mean by qualia. And we're talking here about dancing qualia, which to me is like dancing sensory experiences. Like, does it look like one dress or the other, or do you see the old lady in the picture or the young lady or
No,
whatever?
I don't
But all
think
these
that was
visual
Dennett's point.
experiences.
I don't think Dennett's point was like a two-phase illusion that jumps back and forth between a young and old woman. His point was that what your internal sense of qualia could actually shift back and forth and you wouldn't know it, which just speaks to whether or not the sort of the concept of a definitive qualia is actually meaningful, except
You wouldn't notice that it just changed from one to the other, from 1 second to the next?
I'd have to find his description of the
Well,
experiments, but
I think the word "you" is doing a lot of work there. So if comparing your previous experience is always involving comparing to a memory, and the mechanisms involved in comparing to a memory, then you are completely dependent upon a computational process to verify that the current experience is the same as the previous experience.
Right.
Now, I've said a lot that I think that the key problem here in all of this is making an assumption that there is a current experience. And if we were to throw away that assumption and just accept that all of these things are comparisons against memory, then it's essentially because Daniel Dennett is at some level an illusionist, right? And he's essentially saying, look, there are certain things that you think you can get access to because of primary experience, but that's wrong. It's all being filtered through, a computational mechanism. I feel like,
can I stop you for a second there? Just what does it even mean to say that something is not filtered through a computational mechanism? Like, how else does one get anything?
well, I think a lot of people, like the IIT people or something, they will say that there is a experience first, and that experience is then remembered or put through a computational process. I don't know what I'm saying.
Okay, I'm sorry. I guess it's me again, just being so narrow-minded that I can't even imagine taking the perspective where experience comes first, and that's probably wrong. It's wrong of me to make that assumption. Yeah, good point. Okay, so there is a different way of looking at it.
So I guess there is some way to devise some kind of visual stimulus to test whether there could be two different qualia for the same thing. without having to rely on memory. For example, if you present something on the left visual hemifield, and was the same thing on the right visual hemifield, and if it's the same person experiencing this qualia, but if this kind of illusion is successful in giving this red qualia on one visual hemifield and a different qualia on the right visual hemifield, then that's a direct comparison of this experience without requiring memory.
At the risk of seeming annoying, can I ask the same dumb question again? What's the difference between a sensory or perceptual process and its output versus qualia? Because here you're saying you use left versus right hemifield and you get one result or the other. To me, that's just different outputs of visual processing. I'm not sure where I see this concept of qualia becoming defined.
Right, so that's why I'm saying I'm not sure what kind of illusion it might be, but if you have some kind of illusion where you can elicit from the same visual properties to elicit two different qualia in one person's mind, then this person can report, okay, so I've experienced this strange, dissociation or something.
I'm going to get even more fundamental because I guess I'm just this uninformed. How do you ask someone about the qualia they're experiencing? Can you ask them what is your qualia right now, or what are your qualia?
Right. So here the point is that
how do they know what to say?
So the point is here we are not asking what qualia do you experience. That's going to be a huge difficult issue. The key to this particular experiment is you can just ask them, do you have a different qualia between these two?
Wait a second. Right. You just said you're asking, do you have different qualia? You're using the word qualia again, so you're assuming that the person being tested knows what qualia mean.
Oh, so you can say, do these two colors appear different to you?
So then are we just saying, because if you can ask that and you use that as your measure of qualia, can we just say that experiencing different colors equals different qualia? So qualia is just different sensory perception?
Oh, yeah.
Well, yeah, that's a good point. That's the best we can do, I guess.
In the domain of color perception, yes. I mean, qualia has always been regarded as the perception of different colors. But, I mean, there's all these other kinds of qualia. I mean, we've been talking about pain in this conversation.
Okay, so differences in perception equals qualia. Is this the definition we're going towards?
The difference in perception is qualia. I thought it's an interesting way to phrase it. Because there's no other way to define it, so we just define it by being different from something else.
Okay, thank God.
Where would you put blind sight in that category? Because obviously you are perceiving stuff, but you do not feel like you're perceiving stuff.
That's an interesting point.
So I mean that's to me that's a clear distinction that there is a subsystem in the brain that is our touchstone for conscious experience, and there's a bunch of subsystems in the brain that are still doing what we would call perception, representation, computation, whatever, that are outside of conscious experience.
And to you, Ken, would that, for example, if you were to try to think about this mechanistically, would you say that this is where, say, the perception hasn't entered the self model? And yes, I'm putting words in your mouth.
Yes, although there's a ton of stuff about is the self model really is it making up the best summary explanation of what has happened, as opposed to really getting to the guts of what's in V1. So for instance, you'll swear that you see more of the visual field than you actually see. You'll swear that you see things in a change blindness experiment, and you didn't record those in a way that you can respond to them.
Yeah.
And so the assumption is that there's a self model that is I don't want to even say filling in the details. That is basically putting in a statement that says, "I see the whole visual field," and that's it.
Yeah.
It's like,
Yeah, it's not just doing that because it does that. I think that's a very important purpose of a self model, is to maintain a stable model of not just yourself and what you're seeing, but of what you think is out there and everything else. So I think that fact that it fills in, makes up summaries, is probably absolutely essential for a self model. But that's interesting. So then qualia are more in the domain of the self model, or entirely in the domain of the self model. So it's what the self model is making up about, or how it's construing some kind of summary. That's interesting. Yes, Yunsuk.
Yeah, I guess to add on top of that, I guess the main difference between people with blind sight performing something and someone with normal sight performing the same task, the main difference is in people with blind sight, they're totally uncertain about what they're doing, whereas people with normal sight, they're absolutely certain about these things. So I guess it's not just the self model, perhaps, but how certain are you about the self model?
Mm-hmm.
Like people with blind sight might have some kind of self model, but they're totally uncertain whether that's correct or not.
well, I mean, I would say that the self model in those in people with blind sight is getting the information that they couldn't possibly know what they couldn't possibly walk across the room because they're not getting continual feedback that says I'm seeing everything, I'm seeing everything, I'm seeing everything, and so their uncertainty comes from that lack of feedback, whereas a normal person is in the extreme is just getting a kind of in their self model, in this subsystem that is handling a self model, is saying it's okay, you see everything, it's okay, you see everything, it's okay, you see everything. It's not saying I see that chair, I see the cat on the floor that I'm stepping over. It's just saying I see everything, chill. uh, and so the perception that's actually doing the hard work of walking across the room is still in a subsystem that's outside the self model.
Yeah. So this is interesting now, because we had some descriptions previously of ideas of maybe testing qualia in animals like aplysia. But we're now saying that the difference really between qualia and simply perception is that the qualia have been adopted into the self model in some way, and they might have been completely made up, or they might be partially made up, or they might not get enough feedback, or whatever. But then it should be fairly impossible to test qualia and aplysia, because we're assuming they don't have a self model.
Exactly. And at a larger level, it's kind of okay. So bringing it back to whole brain emulation, uh, the subsystem that is the self model is the touchstone that that should be used to judge whether it worked. It has the kind of meta level information that that has recollections of what it was like to be, maybe even false recollections, but that's fine.
Yep.
Recollections of what it was like to be the individual. And if the emulation matches with that, then is good. Even if V1 has been replaced with a generic V1,
Yeah.
as long as that did not affect the check off of the self model, then it's still good. Now,
But
if
again,
the self model is kind of jury rigged to always say yes, then that would obviously be bad. So there's a little bit of a circular argument there.
right, right. That's like this question about whether you can ask something, well, are you really still yourself? And then it keeps saying, yes, I am this person, but it doesn't mean anything because you don't know what else behind that. But again, I do agree. I think that when it comes to has an emulation worked, it's really at that level, the only level that we're really aware of, that that's where it matters. But it's probably not something where the sort of comparison between what does the original system build in its self model and what does the emulation build, are they exactly identical, but more are they acceptable or characteristic or something like that? Because I'm assuming, again, there's an envelope of what happens there, right? And we probably change this as we grow and over time and from moment to moment and one day to the next, and how we feel and all sorts of other things affect our self model.
I guess this can go on forever. So, Randall, do you have something else that you wanted to ask us?
Yes. Yeah. I mean, I should say I found this to be very useful because we eventually, of course, we have to close the loop back to what are the success criteria for whole brain emulation. And this, even though it's really hard because it's really philosophical and very difficult to pin down exactly what we're talking about, is probably what we have to answer for anyone, because that's where all of the fundamental questions about whole brain emulation and mind uploading come from. And when people say that can't possibly work, it's always about this side of things. So I think it's super important. But because I suppose people are getting tired of the conversation.
Oh, no, I have tons of more stuff to say, but
I would say feel free to say it, because our next, the last topic, because we've combined two discussion parts here about models and about data. They're both involved when we talk about perception and qualia and about building a model and all that. So I think we put those together. So the only remaining discussion topic that I had in mind, not to say that that should be the only one, you can come up with whatever you want, was about lo-fi whole brain emulation, which is an interesting topic, but I'm super interested in anything else that you have to say about this topic.
Okay, so I just have one more thing to say here. So I guess when we talk about the self model, then think about it. What are the tests that we currently do to test whether a particular animal has a concept of self? So the test is typically the mirror self-recognition test, right? So a lot of these animals, chimpanzees or dolphins or elephants, they pass this. But some other animals, maybe perhaps even your cat, may not pass it. So can we say that it doesn't have qualia? So that's one issue. So and even animals like Ecclesia, so do they feel pain? Do they have this pain qualia? So obviously, I guess I wouldn't say that they have the sense of self as in the human sense. Okay, but then the question is, does qualia have to always be associated with the model of the self? So I feel pain versus there is pain.
Yeah.
So in humans, we might feel I have pain, whereas the Ecclesia might feel there is pain. I have no idea what is feeling this, right?
So I put a little comment down here. I think that, see, when you say, does a cat have a self model? I would still put it in a different category than Ecclesia, because I think that a cat probably has a notion of, say, where itself is in space and itself as its body and everything like that. So I think that there are degrees of having a self model. Self-recognition, even though we're taking that as a test right now, there's no real proof that self-recognition is the test for a self-model. No.
That's a pretty high bar, I guess.
It is a very high bar. It's like almost human level self-model. Yeah. Okay, this is cool. But yeah, let's switch to the other topic. Who knows? We might have other comments on this stuff afterwards. Okay, we did that one. All right. So another thing I hope to touch upon, because lo-fi is being proposed all over the place for various reasons that we can also discuss, but I'm really interested in in determining how we distinguish the two and whether or not lo-fi can even be categorized as the same sort of thing, whether maybe it should actually have a totally different name and not be called lo-fi whole brain emulation. And I'm coming with a few assumptions, and the assumptions might be wrong. I'm assuming that lo-fi approaches don't collect high resolution data within the brain of an individual animal or human for which an emulation is to be created. I assume that lo-fi approaches rely heavily on a combination of general models of cognitive processes, general models of brain mechanisms, and externally observable in vivo data like video data, and that they often look to AI implementations of cognitive capabilities as a guide or tool for the construction or derivation of an emulation. This is what I'm assuming, but maybe you guys know better from your discussions with people who are advocating these types of approaches. And so I'm curious about your thoughts on lo-fi distinctions or differences, whether their success criteria are completely different. In other words, are they aiming for different goals? Are they aiming for the same goal but taking a whole different approach? And does that look likely to succeed? So any thoughts that you have on this new phenomenon of people calling what are basically either black or gray box studies with maybe some MRI thrown in, that also call themselves whole brain emulation? Yes, Ken.
Well, I would just say that and I've said this to them as well, I think it's incredibly, incredibly dangerous because this is the kind of way that you would get an external behavior like a human being, but either no one's home, philosophical zombie level, or something completely alien is at home, but you've matched the external behavior very well. I don't think if you're building from the ground up, you would never stumble into something that works, but works in a fundamentally in a way that doesn't capture the inside. But I think this lo-fi approach is basically bumbling into the philosophical zombie apocalypse.
Okay, so two quick comments. Some of the approaches do propose to use, say, MRI, for instance, as some way of looking at the inside and seeing what activates in relation to what else. So they might claim that even if they're not capturing the details, because they're capturing that level, then you're getting some insight into whether the insides are operating correctly. I don't know how far to take that.
I mean, the thing is that I could never, and I've drilled in on this with some of them. I could never see how that was enforcing a causal set of relationships as opposed to just adding another constraint. So it's like, oh, we are both matching the outside and this MRI. Okay, fine. But that doesn't mean that they're related in any way. You've just matched them. And I think this is a fundamental thing about any of these black box approaches.
Yeah. So that it's not a causal relationship. Okay. The other big question that I had related to this is, so could a lo-fi approach, and do they even want to, aim at what we just determined was most important, say the self-model part of what's going on in a mind? Like that self-model, is there any way where lo-fi can attempt to capture what the self-model is doing and maybe qualia? And maybe that relates back to our discussion of whether you can test self-models in aplesia. In other words, can you look at behavior to test qualia? Richard.
I don't know if this adds to the conversation, but Anthropic has been like running experiments on the language model. So the language models passed the whatever Turing test in 2023, right? But we say that the Turing test is kind of a weak experiment. Need a better experiment for that, right? But we don't quite have one yet, right? And so Anthropic, what they're doing is they're doing this quote unquote chain of thought reasoning, and they're saying, okay, we're gonna prompt the language model to uh, have line up their thoughts and then output an answer. So the researchers are trying to read the thoughts of the language model before outputting that answer. And so I don't know if this is like answers a part of the consciousness question as well, but I don't know. It's just something I've been staring at for a while.
Well, I mean that does seem to at least acknowledge what Ken was saying about causal relationships and try to look at causal relationships, because if you're figuring out a chain of reasoning, you're looking at the causal relationships in a sense. You're going from one thing to another.
Yeah. And of course, LLMs are statistical machines, right? Yada yada. They're not saying actual thoughts. Yeah.
This has previously been said about human brains that we're statistical
Oh.
machines. And at some level, of course, it's always true. If you have a neural network doing something at some level within that little box of a little circuit, it's doing something you could describe as a statistical machine.
Yeah.
Um,
Well, I guess this is where like meditators can come in, and they're like, okay, yeah, I'm gonna focus on myself. I'm gonna think about the self. What is the self? What is my self saying? Uh, so I mean, I guess you would have to meditate yourself to see what the other self is trying to say about their selves.
yeah.
So yeah, it gets to a whole circular thing.
Okay. So
I
then
think.
what we're saying here by comparing lo-fi and what we're normally talking about, or we could call it hi-fi or classical or whatever, um, is what we're basically saying that we really believe that setting our success criteria in a way where we need to do evaluations of circuits or even smaller, where we need to go down to some some component level that is much smaller than what lo-fi would look at, that is that we can somehow give assurance that that produces the results we're actually looking for, that we're saying, for example, you know how in the success criteria part we discussed looking at evaluating success criteria by using metrics at different scales, looking at different scales and trying to match them up across scales, that was important. And I don't think we ever made this really formal or really described clearly why that is, or what our how we substantiate that. But that's really the main difference, right? We're saying if you're doing lo-fi and you're looking at the whole box, then you can only do tests at one scale and not at other scales to compare if things are the same or similar enough.
Mm,
So we're making an assumption or postulating that doing this multi-scalar comparison down to some scale, which we're still trying to determine, that gives you some level of assurance that you're producing the right kind of internal representation, the right kind of self model, the right kind of qualia. This is all very vague still, I have to say.
yeah, yeah. Cause I was looking at Keith's stuff, and like I was looking at the Mitzinger stuff, and like that kind of philosophical like brick wall or whatever. You guys were kind of hitting that over and over when I was listening to you guys, and so like I don't. There's like a what is it? It's like what is it? Why do we eat cows and not dogs? Okay, it's because there's some sort of humanness aspect. Okay, no, because what pigs are kind of smart. They're smarter than dogs, I think. And so okay, is it like the empathy? Are we trying to look for the empathy levels? Like is that our deciding factor? Like the humanness human yeah, I don't yeah. I don't know.
Yeah.
Yeah.
Right. So I have some, I guess, maybe answers and comments to Randa's questions about this self-model. So I guess the self-model, what do we think about self? Right? And I think one really important requirement is that this self-model should be attached to some material thing that it will call self. like a body or a robot, or even some kind of a cube sitting on top of a table, right?
Mhm.
So I think that it cannot be totally in the cloud. So that might be one thing to consider. And then when we think about this low fidelity whole brain emulation, I guess we need to answer this basic question. Who is the audience of this whole brain emulation? Is it the person being emulated, or is it those who survive the death of this person? Right. So if it is the latter, then we have much more wiggle room. Right. So it being lo-fi might not matter that much. For example, there was I saw an article maybe a couple of years ago
Mhm.
when someone fed in her entire diary from her childhood into a
Yep.
language model, and then now started a conversation with this fine-tuned language model. So this is like the lowest fidelity whole brain emulation in some sense. And she was really struck by how accurate the answers or decisions made by this chatbot would be.
Yeah. Okay. So that's very interesting because it kind of feeds into a thought that came up. It's not the first time it came up. It comes up all the time when I'm thinking about different ways to, I guess, to do system identification and to reconstruct something. You can either do that by observing behavior. You can do I/O, like you're basically looking at a black box. You're recording a bunch of things, whether it's neural activity or video or whatever, a diary. Or you can look inside, reconstruct architecture down to some level. This is always where we get into trouble, and because at some level you're always doing the I/O matching. And then you say, because I've looked at it in that much detail on the architectural level, I can say things about this system that maybe just looking at the I/O can't tell us, because you either haven't observed for long enough, you haven't captured everything, or you haven't been able to capture things that you have never seen any evidence of, because the particular I/O that has happened in the lifespan has not yet touched upon certain ways that the architecture can interact. So that's one of the things I always worry about if people are like the hippocampal model, that the first thesis, which is entirely dependent on just input and output recordings and then making a model of what's in between with some architectural constraints based on typical architecture of the hippocampal CA3, CA1. The problem with any of those methods is that you are building a system based on what you've observed, not based on what that system might be able to do to some degree. Now, you could say that if you've watched long enough and if the system is simple enough, then you'll capture everything that's inside of it. But if it has a lot of variables, it's very likely that there are some interactions you've never seen and that perhaps you won't predict by just looking at input-output. So the lo-fi model, to me, has that risk that even if it captures current behavior and even causal relationships as they currently happen, say causal reasoning that you can observe, that it may not be capturing how the system would actually evolve presented with new information or over time, so that it diverges in some important way. But then a person could argue that doesn't matter, because any evolution of yourself from here on outwards is okay, is correct. All of it would still be you. It's just that, because you don't know what you're going to evolve into or what sort of behavior you're going to exhibit in the future. So why would you judge yourself based on that? So it's a bit of a weird constraint or criteria to have, but it's sort of the thing that pops up when I think about those two different ways of looking at a system and trying to reconstruct it.
Yeah, so I guess the really interesting thing about this diary experiment was that she fed in the diaries from her childhood into the language model, and then presented it with a situation in her current life. So what would you do if this situation arises? And actually, the answer from the language model was pretty accurate. So that was the interesting thing.
That's interesting. Yeah, because there's this other possibility that we're being overly like we're assuming too much about the fidelity of our self, our self model, or our entire brain and our mind, that we think too highly of how detailed it is and what we can, how much variance there is. So maybe it's quite predictable. Maybe a lo-fi approach can capture everything about you, um, because there's just not that much that you can expect. I don't know. Um, I'm reminded of, Ken, how you once told me that you think that, say, I don't know if it was about the self model or the consciousness or whatever, that seems like a really flimsy little layer on top of our memories. That the things that are different between us, let's say if you're sad or I'm sad, those differences are much, much smaller than any differences that are driven by our characteristic sort of associations with things because of memories we have. So this layer that people are so concerned about, the very, I don't know, I'm not going to say qualia again, but the variances there between people might not be that big. It might be a really small model that you need to capture. So perhaps the thing that people think they're concerned about when they say this is the most important thing about whole brain emulation might not actually be a big deal.
Yeah, I'm thinking of some of the statements that Michael Shermer has said using the terminology that I put forward, POV self versus MIM self, and he's like going to the mat that POV self is the thing that is important. You know, this instantaneous personal experience is that's the thing that we need to. That's who I am. And yeah, I mean, the essay that I wrote on this was kind of to abuse people of that idea, and it certainly did not did not work on him.
But does it still work on you?
Uh, I I I think it does. You know, there is an abyss that I am worried that will consume me at some point. If you think too far in this direction, you come up with the idea that there is no purpose to whole brain emulation or anything else for that matter. Uh, so you know, I am still searching around for this. This alternative, because I think is what you're saying. You know, it's like this instantaneous. What was I saying? It's like if you're instantaneously um, experiencing a car accident uh, versus somebody else instantaneously experience a car accident, the um, the representations in your brain are probably almost identical at some level. At some level that we can compare these two, they have nothing to do with the individual. And so the idea that those are the things that need to distinguish us is kind of ludicrous, and you're left with you know, a memory based alternative where what we really care about is the unique history of a person's life.
Yeah, I've
Uh,
already found that to be fairly convincing. Um, but then I guess the question when we're relating this to lo-fi is, do we think that lo-fi could capture our memories?
I mean, the thing that I come back with lo-fi is just there are uh, a billion different ways. Correct me if I'm wrong, okay? But I can imagine that there's a billion different ways to get the same external behavior. Uh, um, and some of those ways of getting the same external behavior is to literally create somebody who's pretending to be you, that understands that they're pretending to be you,
Mm-hmm.
right? And they could be just fantastically good at pretending to be you, but they have their own conscious experience of pretending to be you. And we certainly don't want to allow that through. And if it's truly a black box, then doesn't that open up the possibility for just a good faker of
Yeah.
being you? I think that that's also vanishingly small. I think that, more likely there are ways to pretend to be you that are so alien that we wouldn't even want to ascribe consciousness to them.
You mean like a lookup table?
Like a lookup table. Yeah, yeah. Although it would be lovely to go to the mat and say, okay, why isn't a lookup table sufficient to get even a self model? At some point, aren't these neural networks? Can't they be characterized as a lookup table? I mean, they are just
Well, that's
yeah.
right. That's where, to me, it comes back down to the question of scale and what that means. Because, as I was trying to say earlier, at some point you're always dealing with a lookup table or a statistical model or whatever. So if you're doing the model of the neuron or the model of a neural compartment or an ion channel, somewhere you're basically making a function that's a model that is, in a sense, almost like a lookup table or some statistics. And then at some point you have to wonder, well, isn't that eventually what reality is like down at the mechanistic level? But that's aside. So it's really, at some point it becomes that. But
My
what we're
my
really
only
arguing about is the level, really, when we're talking about our way of looking at whole brain emulation and lo-fi approaches, right?
my only uh, argument for bringing this up was to basically say that as a reader,
Yeah,
I would want somewhere within this document a a clear statement of how this particular scale separation is being looked at, and why it's not being chosen essentially arbitrarily, that there really is a, this is the final determiner of whether a brain emulation was a success or not. And if that comes down to, there's uh, a short discussion of theories of consciousness where at least some are crossed off as being nonsensical or too vague, and a few are kind of like, these are reasonable ones to assume for the sake of discussion, realizing that they will be supplanted by something that's less vague in the future.
right. I think I agree there. That's why I started out this whole session with some philosophy, because there are some ways of looking at things that I find are more useful, and the Metzinger model keeps coming to mind. Um, but yeah, making a final decision about scale clearly, and then why that would exclude lo-fi, for example. In the end, gosh, it feels like we're back to the self-model thing, that's somehow what we think is really important, even though we just said that parts of this could be a pretty flimsy layer, the instantaneous part, but that's temporal rather than resolution in space.
I mean, at some level, we're going to, so, if we went back 200 years, 300 years or something, there was this concept of what life was.
Yeah,
And, and now that concept of that, whatever that concept of what life was, has no bearing to what biology really is, what we know it to be, right? It's like, yes, it is just chemistry, right? But it has, but that doesn't mean that the value of the difference of life versus non-life has been lost. It's just been explained. And so the hope is that we're going to get there with consciousness. that consciousness and self are going to the kind of concepts that we are just really wedded to. The very idea of saying, look, we're going to upload somebody's mind, we're going to emulate their mind, will probably seem silly at some level, but hopefully it will be replaced with something that also makes that makes sense, and at the same time that will not render the whole project meaningless, right? It's not like us understanding that life is chemistry has diminished life, because it's so complex chemistry that we can appreciate it.
Yeah.
The whole brain emulation is hopefully going to show how really unique each individual is. That this unique individual um, is a part of the universe that really should be appreciated as a whole, and that allowing them to continue in an emulated form is a really important thing.
Hmm.
It's not just that we've got all these philosophical zombies going around that have come up with this bizarre idea of whole brain emulation so that they can continue their philosophical zombie existence into the future.
So one thing I wanted to add to that is that, so you were talking about how we change how we talk about things, like how the discussion of what life is has changed completely from what it used to be. And I can say that just personally, I don't know that's where things are going to go. I find it much more comfortable to talk about a self model and what is a self model. How do you create a self model? How do you recreate it? How do you reconstruct it? How do you know that it's a good match? Blah, blah, blah. I find that a way more comfortable thing, much more concrete to talk about than, say, talking about consciousness as a thing or qualia as things or all of that. So for me, that way of looking at things just feels much more, I guess that maybe it's just because I'm used to thinking in a certain way, that feels more comfortable. So say I would almost be at peace just trying to figure out a way to do whole brain emulation where you generate self models that are very similar to how self models are generated in the brain. And then not worry too much about what that means in terms of there being consciousness and qualia and all of that. That's just me, but that's sort of my feeling there. Then, of course, we can flip that and say, okay, if we care about intelligent beings that have brains with the kind of cognition that we have and with self models, then we might not care about more than that. We could say, that's good enough. Maybe that allows people to continue living because maybe that captures everything there is about us. And I feel strongly that it may. Or maybe not, but then still whole brain emulation would have the purpose of ending up with a species that is substrate independent and has all that cognition and self models. And why is that any worse than whatever we had before? If you can point your finger at exactly what's missing and why that's important, then I'm all ears. But right now, that all feels very vague to me. So anyway, that's a controversial statement, but yeah, if you're thinking about ways that this could develop the way the discussion about life developed, that's sort of where my head goes.
And I think that yeah, go ahead.
Okay, so I guess so this kind of discussion about scale and everything, I think we need to think about emergence. So reductionists don't like this word, but I guess it's a good concept to just frame our discussion. So at what level, when we emulate, would it give rise to this emergence of some higher level phenomena, something like that?
Yeah. And so I tie that kind of discussion of emergence into the concern about what do you need to capture about the underlying components, so about that system that's there, so that you can say something about the future evolution of the system, the path that its thinking takes. Or, and Keith is gone, but that's fine. The To me, that's very similar, because emergence isn't just what capabilities are there in the moment, but also how does the system develop over time. And so what behavior might you observe in the future, because that behavior is emergent from the system. So the emergence to me is tied to some degree to the scale at which you reproduce the system and whether you're going to get the same emergent behavior. Of course, this goes right back to the whole conversation about the goals of emulation, because, for instance, if we just look at emulations of computer systems, you could say, well, some emulations, their emergent properties are that they produce the same program output, and other emulations will have the emergent phenomena or properties that you will also be able to tell that certain parts of the system are active at a certain time, and that there are temperature gradients and whatever else you can see in the computer system. So the types of things, the types of emergent things that you care about goes right back to the question of goals and success criteria.
Yep, I agree.
Where I think what we've come to in this study session is that we believe that self-model is a very important emergent system that is crucial to whole brain emulation the way we talk about it. But yeah, not sure how far we were able to drill down to other things. And I do think there is something to be said here about lo-fi and the emergence of such things. I don't know if lo-fi even looks at the emergence of a self-model or how they would test that. Yeah.
Well, I think very briefly. I think we've seen large language models be very easily misinterpreted as being able to reason about particular tasks, and it took very particular examples to figure out that, they are not reasoning about those tasks. They're actually that you can see flaws in their not flaws. It's that they're not doing the type of reasoning that we are assuming that they are. And so that should give us real pause that there aren't really, really good black boxes that will seem like there will be a self model in there, but there won't be a self model.
Mm-hmm. Yep. And then how do you test that? What are the metrics? I guess if you see, I'm getting back to this thing where I'm saying what we're assuming going into this is pretty much that if you do tests at multiple levels, at multiple scales, we're saying, okay, because I've tested at all these different scales, my assumption is that the self-model being generated is the same or is very similar. So we're saying that we're not really pointing to a particular test for the self-model to say be able to compare between one that seems to be there versus one that's really there. We're not saying here's a test for that, but instead we're saying we are assuming that because we're doing the reconstruction at a much higher level of detail, that the chances that it is the same self-model are good. Is that correct? That we're making that assumption rather than that we're specifying a test?
I mean, I think yes, and I think that it's a good assumption.
Okay.
Because it's like I could see where it would fail, and I could see where it would succeed, but I can't see where it could accidentally look like it succeeded. If you're coming from the bottom up.
I wish we had a test rather than just an assumption, but I do agree with you. I feel I'm more confident about it. Richard?
I wanted to bring up some stuff about fruit fly, I guess, and I don't know if it's relevant. Let's
Sure,
see.
go ahead.
So so we're measuring neurons of fruit flies, and then we are able to tell the direction based on the neurons,
Mm-hmm.
right? Um, would like one experiment be like, I don't know, putting the mirror test and then like seeing which neurons lighting up, and then is that a self model? I don't
Which mirror test are you referring to here?
I don't know. I saw one post on it on the internet in 2009. So I don't know if it's even real.
Yeah, I
But
noticed that in the chat you wrote fruit flies pass the mirror test, but I'm not sure if we're talking about the same mirror test. You know
yeah.
the mirror test that they talk about in chimpanzees and dolphins where they can recognize themselves? Like they'll notice if there's a dot on their nose or something.
Oh, yeah. Then I don't know how deep they get into that particular mirror test, but I don't know. I guess that could be helpful? Question mark for I mean just like generally testing self in a fruit fly and then seeing neurons lighting up and emulating that, and would that be self? Uh, yeah, that was it.
Mm-hmm. Yeah, I think testing whether neurons light up, that's really the granular level that we're usually talking about. And the mirror test is very much an IO out, black box test of and the organism recognize itself. Yeah.
Mm, yeah.
Okay, we have actually reached the top of the hour, so the session is formally over. I think there's still a whole ton to say here, and really, you're right, Ken. We would like to see certain things in this roadmap, some statements, like reasons why we choose a certain scale, why we would say that one theory is more reasonable, better to use than another. Those are the sorts of things that do belong in a roadmap as some sort of guide. What I'm still missing and trying to work on is trying to collect a good set of papers that show some of the things we've talked about. And I would love it if we can exchange papers if you have any good ones you'd like to point to, or if some come up. So I'm going to do something about that to try to arrange some channel where we can exchange and collect these things. Also, Logan and Maria, they've been championing building this roadmap in a relatively open way where we work on documents, but then as soon as we think they're about draft ready to show with people, we're putting them on a website. That website, we've actually gotten the domain name wholebrainemulation.org for that. Yeah, so somehow we're going to try to get a lot of input from people as we go along. As you probably can tell from this session, I was trying to cover quite a bit of ground and trying to be relatively detailed, but there are still so many areas where things seem a bit vague. And tying anything into literature is probably a really good avenue. So that's one thing I would love to ask for is please send me your ideas on good references and anything I should look at to better understand points you were making, anything of that nature. Are there any further comments or things anyone wanted to say or discuss? If not, then thank you all for being here. I thought this was very productive and good. It's definitely not going to be the last time that we do sessions related to the roadmap. More like the first time in this cycle. So thanks, everybody. And
Thanks,
yeah,
Randall.
see you
Thanks.
around.
Good to see you. See everyone.
Bye-bye.
Bye.